Hi, I'm Stuart Roberts from NDF Research, and with me is Dr. James Garner, who's the CEO of Kazia Therapeutics, which has just reached a great point in its clinical development, initiation of the first trial site in the phase 2 study in glioblastoma of GDC-0084. Now, James, it's fair to say that glioblastoma is a fairly bleak condition in terms of new drugs that have come along to treat the condition in the last 15 years. We've had temozolomide. We know that radiotherapy works in some instances, surgery, and one single agent that's been around since forever, and that's it. You could be the next big thing.
So we certainly hope so. We think we've really got a great shot here with GDC-84. The challenge with glioblastoma, as you've noted, is that there really hasn't been much progress in the treatment of this disease for 15 or 20 years.
Right.
The current treatment paradigm is that patients receive surgery, they receive radiotherapy, and then they go on temozolomide, and that drug is now about 5 or 6 years off patent. It's been around for some time, and it definitely works, but it only works for about 1/3 of the patients.
Right.
And so we know that for the other 2/3 of patients, there really is no generally accepted pharmacological therapy that offers benefit. And so that's the target for us. That's the gap we're trying to fill with GDC-0084.
Right. Now, the FDA granted you orphan drug status for glioblastoma. It's a rare disorder. Only about 12,500 Americans get it, albeit, unfortunately, prominent people like Senator John McCain. What's the commercial opportunity for the drug once it goes live?
Well, temozolomide, before it went off patent, was a $1 billion a year product,
right,
mostly off glioblastoma
right,
cells. And so that gives a sort of indication of the landscape here. And if we look worldwide, there's probably somewhere around 130,000 patients a year diagnosed with glioblastoma, about 10% of those in the US, about the same again in Europe, and then the rest elsewhere. So this is still a pretty significant commercial opportunity. And although we think of glioblastoma as a rarer cancer in the context of, say, lung cancer or breast cancer, the reality is that we're breaking those cancers down now into
right,
subgroups. You know, for example, one of the areas of activity in lung cancer has been with ALK inhibitors,
right,
which only really treat about 2% of the lung cancer population, but there's still 3 of them on market, and it's been a very viable.
And they've all been very successful drugs.
All been
right,
extremely successful,
right,
exactly.
Right.
So there's real opportunity to make commercially viable products that are targeted to quite specific populations.
Right.
So we're confident that we've got a very meaningful commercial opportunity here.
And obviously, this is an important time for you now. You in-licensed GDC-0084 from Genentech in October of 2016. Here we are 18 months later. Now, my impression is you've worked at lightning speed to bring the drug to the point where we're initiating this phase 2. Talk to us about what you've achieved over the last 18 months.
Yes, sure, thank you. It's always easy to underestimate the amount of work that goes into setting up a major
right,
international study. You know, these are huge projects, and for sure we've been extremely busy since we brought the drug on board. There's been a lot of regulatory housekeeping work to do, and transferring the drug over from Genentech, so that's been quite important. We work closely with a consultancy in California to interact directly with FDA. We've had to manufacture capsules for use in our clinical study.
Because this is orally available once a day.
Right.
Yeah,
that's right. So it's a daily dose drug, right, and we were very fortunate that we received about 48 kilos of material through the transaction with Genentech, but it was literally in a powdered form. We needed to manufacture that into capsules, which is always a bit of work in itself. So we've been through all that manufacturing work. We spent a lot of time talking with the clinicians and with our advisors about how to get the study design right. We really want to learn from past mistakes in glioblastoma and make sure that we've got the best shot on goal.
Right. So one of the advantages you get moving into this current phase 2 is that Genentech laid the foundations for a pretty competitive drug in the space, given other agents that have been tried in glioblastoma. Talk to us about some of the aspects of the drug that you have in-licensed from Genentech, which you found helpful.
Genentech did a really great job, actually, with the early development of this drug. It's an unusual drug in that it was developed from the ground up as a brain cancer drug. Most of the drugs that have come to glioblastoma have really been breast cancer drugs or lung cancer drugs. where the company has tried to sort of test them out in brain cancer.
Right.
Genentech approached this very differently. They started with a blank sheet of paper and said, how do we make a great drug for glioblastoma?
Right.
So first of all, they identified the PI3K pathway as their target. And we know that the PI3K mechanism is disordered in about 85 to 90% of glioblastoma patients. So it's a very, very attractive target for this disease. Then the second challenge they set out to address is that drugs to treat brain cancer need to get into the brain. They need to cross the so-called blood-brain barrier, and most cancer drugs don't.
Right.
So designing this drug, doing the detailed chemical engineering to get it to cross the blood-brain barrier and get into the brain was really a huge focus for Genentech, and they did a magnificent job there in the chemistry of the drug. And then finally, a lot of the early development was really very rigorous. They did a great deal of animal work. They ran a very sort of substantial and well-designed phase 1 study in 47 patients.
Yes.
So we've had the benefit of all that data, all that experience with this drug, both in animal studies and in human studies, that we've been able now to feed into the design of the phase 2.
Not only that, but the study sites that Genentech obtained were pretty prestigious. We had MD Anderson, if I recall, UCLA. So a lot of investigators who may be talking to you now about participating in your current study possibly have seen this drug before.
Absolutely, and we've been really grateful for the support we've had from some of the investigators who participated in Genentech's study. It's just always terrific to be able to work with sites with that sort of caliber. And we're hoping that some of those individuals will continue to participate in the phase 2 study as well. And they've certainly given us great input. And we've had a great deal of support from Genentech as well. We've been really grateful for their very thorough handover and for their clarification and support and guidance as we've been designing the phase 2.
Now, you've got an interesting trial design. You've got a control group effectively in this study, based on the fact that not everyone will respond to temozolomide. So you can pick the temozolomide non-responders, and this could be a treatment option for those people that they've never had before. Talk to us about that aspect of the design.
Yeah, Stuart, you're absolutely right. It's usually the way with a new cancer drug that it ends up treating patients right at the end of the line. Your patients who've failed every other drug known, and then they come into a clinical trial. And naturally, those are the hardest to treat patients, the patients for whom no other therapy works. Glioblastoma is almost unique in that it gives us an opportunity to target patients in a first-line setting, to get patients quite near their time of diagnosis.
Right.
And the reason we have this opportunity is that the approved therapy, temozolomide, we know only works for about 1 in 3 patients. The other 2/3 of patients, although at the moment they receive it still because there's really no alternative, we know that it's offering them almost no clinical benefit. And so it gives us the opportunity to position our drug in a head-to-head setting against an approved standard of care, but in the knowledge that that standard of care offers almost no clinical benefit.
And selecting those patients is a simple blood test, right?
Well, it's a test on the basis of it.
Right.
That's right, on the basis of a tumour sample, and almost all of these patients have surgery early on in their treatments. We have access to a tumour sample. We can test to see prospectively whether they will ever respond to temozolomide. We don't have to try the drug and wait for it to fail. We can tell in advance.
Right. Now, you've got an interesting, I'll call it partly adaptive trial design. Talk to us about the opening phase of this study and how it will be different from what follows.
Sure. So we're going to start the study with a dose optimization piece. The Genentech phase 1 study was actually in a slightly different treatment population. It was really in very end-of-the-line patients who'd had, on average, 3 prior lines of therapy. So these are tough patients to treat.
And interestingly, there was hints that we got disease stabilization in those patients,
Absolutely.
Right? Yes.
So even in that really tough population, we got some good signals of activity.
Right.
But we're going to be using the drug in actually a much healthier, earlier stage population. So it invites the question whether we can actually dose a little bit more of the drug and dose in a slightly different way. So we're going to invest a little bit of time and effort in the first sort of 9 to 12 months of the study just to answer those questions and make sure that we've got the optimal dose and the optimal way of administering the drug. And then the results from that part will just seamlessly segue into the main randomized controlled study against temozolomide. So it's really sort of a couple of moving parts to the study, but we think it's a good investment of time and effort to just make sure that we go into it with the best in the best position we can.
Now, obviously, you're still in the process of building out your network of trial sites, but will there be any in Australia?
Absolutely. So the initial part of the study, this dose optimization component, will probably be US-based.
Okay.
Just partly for efficiency, cost management, so on. It'll take place at a smaller number of sites, probably about half a dozen. But as we transition into the main randomized control part of the study, we'll expand the pool of sites quite substantially. And our intention is to include centres in Australia and also probably some in Europe as well. So this will be a pretty fully fledged international study,
right.
and I think we're really excited to be working with clinicians right around the world who specialise in this area.
With details to be posted on clinicaltrials.gov, right?
Of course,
okay.
Of course.
Okay, so for those of you watching, clinicaltrials.gov is a US government website which puts clinical trial details on the web. It's a great resource for patients and their caregivers looking for clinical studies. When I worked in biotech companies, and James, this would be your experience, patients often call up saying, how do I get in?
Absolutely,
right.
Absolutely. And we're receiving those kinds of communications already from both patients and clinicians. You know, the heartbreaking thing about glioblastoma is there's so few treatment options for patients. There's been so few new drugs
right.
in recent years that I think that there's this enormous desire and demand for new treatments. So I think we've had a lot of that sort of interest so far.
Sure.
And, yeah.
Now, when I was doing my work on Kazia, one thing that was important to me, looking into the mechanism of action of the drug, we've got a PI3K inhibitor. Now, some of the more knowledgeable investors might be wary of a PI3K inhibitor, given historically some of those agents have failed. Talk to us about what's changed in the PI3K treatment landscape, if I could put it like that, and how that's relevant to what we're doing here with GDC-0084.
Yes, sure. So PI3K inhibitors are a class of drugs where people have been looking at for a number of years. And I think like just about any new class of drugs, it's a process of learning how best to
right.
use them. I think we've either tried to use them in very aggressive disease where the drugs haven't really got as much traction, or we've tried to use them in very, very toxic combinations which have just been intolerable for patients. And we think we've really learned some lessons here from that prior experience. We also think our drug is probably pretty differentiated from most of the PI3K inhibitors out there. Partly it crosses the blood-brain barrier, which is nearly unique in the PI3K space. It's a pan-PI3K
right.
inhibitor. It hits all the main forms
right.
of PI3K.
Right. So there's 4 different types of pan-PI3, but possibly
exactly.
other developers have hit the wrong ones, right?
Yeah. Exactly,
right.
That's right. And we know now that some of these PI3K forms are really just relevant for hematological malignancies, blood cancers.
Right.
But our drug, we know, hits the forms that are relevant to solid tumours. So I think that's been really important. And I think certainly the data, the data we've seen across the whole field is that these drugs behave very differently. And we know that ours so far has proven very safe in the human trial experience. So I think we're really encouraged that we've got a drug that's really quite well differentiated in the space. And of course, there are 2 approved PI3K inhibitors on market.
Right.
So this is a really well validated target. We know that we can produce.
And in the case of the second of those from Bayer, that's exactly. the same class as yours, pan-PI3K.
That's right. That's right. It doesn't cross the blood-brain barrier, so it'll never be a brain cancer drug. But that's right. Alicopa, the Bayer drug, was approved September 2017, and has really helped to. Yes, the second drug in the class has really helped to just focus attention on the fact that PI3K is a well-validated target for cancer drugs.
Now, coming back to your current study, your primary endpoint in the study is always important. Here, it's progression-free survival. It's fair to say then we can reach a data readout point by cancer drug development timelines in a reasonably swift fashion. Talk to us about what happens after you get to the end of that recruitment process and then are able to read out what the data has to say.
Yeah, sure. So you're absolutely right. We're using progression-free survival as our primary endpoint, and this is a measure of whether the disease recurs or the patient passes away. So it's a kind of composite measure, and it's an endpoint for clinical trials that's really gained a lot of traction over the last few years. And in fact, FDA's own guidance documents indicate that this can be a very viable primary endpoint for glioblastoma trials, and it's certainly something we discussed with them when we met them last year. We made it clear this was how we were going. So, as you say, the advantage of looking to progression-free survival as an endpoint is that it's much quicker than looking to overall survival, which is the classic drug development endpoint. Patients with glioblastoma typically have a median progression-free survival of something of the order of 6 months. So it means that we get a much quicker readout on our study, and we expect to have data coming out probably about 12 to 15 months after we complete recruitment to the main randomized controlled part of the study. But we will have some interim checkpoints along the way, so if we're getting an answer sooner, we'll be able to call it at an earlier stage.
So it's fair to say that the agency has given you the maximum possible flexibility. If the drug is showing great outcomes for patients, we could move this forward fairly quickly into a marketing authorization type setting.
Sure, that's right, and of course with the caveat that the FDA is always cautious about committing to these things in advance. But absolutely, I think there's ample precedent here, and certainly our hope, our ambition for the drug is that after completion of this study that we've started today, we can look to take that data to the FDA and have a discussion with them about accelerated approval, which is this mechanism whereby FDA will sometimes approve a drug without waiting for a full phase 3 clinical study. And so we've done everything we can to really design this study to optimize it for the possibility of accelerated approval. Obviously, no guarantees till we get the data, but FDA has been really pragmatic in general for diseases like this, where there's such a paucity of existing treatments, where the prognosis of the disease is so dire. FDA is really very rational in wanting to make drugs available to patients as soon as there's a good reason to do so.
So finally, we're talking 12 to 15 months from recruitment. We're likely to have a data package that will give us a decent readout. What kind of time are we looking at to recruit the necessary patients in this study?
So the rough breakdown of time is going to take us about probably 9 to 12 months to do this dose optimization lead-in component. It's probably going to take us sort of 12 to 18 months to fully recruit the main part of the study, and then probably somewhere around 12 months after that. So we're looking at about a 2.5 to 3 year timeline for the study end to end. Now, as I say, we will have opportunities to check in along the way. So if we're really, we've designed it quite conservatively so that really even a modest treatment effect we can detect in the study. If the drug meets the kinds of expectations that we have for it, we may get a readout earlier, and so we'll be attentive to that, and certainly be sharing some of those checkpoints with investors over the course of the study.
Well done. The Kazia team have done an outstanding job in bringing the project to this point, and now it's reached the next stage of its development. So hats off to you and your team, and good luck to you in the next stage of the journey.
Thank you. Thank you.